All right. Well, we are here today with Dr. José Ostaiza, who is an long-term CrossFitter and physician, believe it or not. So, Jose, I've known for a number of years now through different gatherings and events. We've crossed paths and chatted over the years. And recently, Jose was able to publish a couple of articles looking at the impact of high intensity interval training and other modalities too on basically health span and lifespan through the lens of epigenetics, which is fascinating. And so we're going to dig into those today. But a little background about Jose first. So Jose is from Ecuador, was a physician in Ecuador for a very long time before moving to the United States. And when foreign physicians move to the United States, typically we have to redo training. And so you're in residency right now in Texas, correct, in family medicine again, despite having practiced, gosh, for almost a decade already in Ecuador. And this is why some of our foreign physicians are some of the most educated and experienced by the time they come to the United States because they've already been practicing for a very long time. So it's an honor to have you here today, Jose, to share kind of your recent research and what you've been working on. And in addition to being a physician, you are a CrossFit coach. You are an L3 as well, correct? And so you've been CrossFitting, I think, since 2011, if I'm not mistaken. Yeah, so that definitely qualifies as been around the block. So you've been doing this for a long time, despite looking like you're 20 years old.
That's the epigenetics that we are going to talk about.
Yeah, that's fantastic, man. That's fantastic. So, without further ado, I'll hand the floor over to you. I may kind of interject with some questions as we're going along to kind of have you dive deeper on things or paraphrase things, but the floor is yours, my friend.
Okay. So the reason that we are here today is to talk a little bit about epigenetic aging and high intensity interval training. I will try to connect the dots between what is the linking between them. And I would like to start with why did I start doing this research? CrossFit was bragging about, or always brags about, that makes us younger and promotes longevity. And I was like looking for all the data that shows, and we have some reasoning behind the VO2 max and how it gets improved compared to other people, and also different biomarkers. And if you have studied the level 1, probably you know the continuum of sickness, wellness, fitness. But I wanted to get something more like data, more observational. If you are in the Occidental research, you know that the thing is like, show me the data the whole time. And I wanted to see how it's at the DNA level, measure the longevity, and if CrossFit really works on that. And how there is also a current that some scientists and some doctors are treating the aging as a disease. So with those things in mind, I started to thinking how CrossFit actually promotes longevity, but also brings back from the aging, retrocedes, like comes back and doesn't let you get on that disease in this same continuum on sickness, wellness, fitness. And the first thing would be like to define what is the epigenetic, what would be the aging, and what would be the high intensity interval training, the effects on that. So if you've been here for the last 30 years, you remember that 30 years ago was the project, the Human DNA project. It was like, we will never be able to read the whole human DNA. And then 20 years ago, it's like we finished reading the human DNA, and there was this new process thinking that we are going to be able to manipulate the DNA, thinking that it was only like this, like reading a barcode and getting whatever the results is. But it's very well more complicated than that. The DNA, we realize that it's something may happen to the code, the barcode, something happens to the beam, something can happen to this reading gun, something happened to the person that is processing, and even getting the product from the DNA was not just as straightforward as reading and getting it. And this is where go epigenetics. When I ask someone what do they know about epigenetics, or when I say genetics, I would like to ask, where do you wanna? I want to see. I want to know what do you think when I say genes? And some go from micro, some go from micro, and this can be very overwhelming if you just see it without an explanation. Some people may say, when you say DNA, I think about the double helix that goes up, or some just say the chromosome. Micro or macro, I will go that the DNA double helix wraps around these histones, and then these histones get together, and then they keep wrapping around, and then until they form these chromosomes that everyone think about, the X and the Y. And the epigenetics pretty much studies which one is get read, which one gets expressed, translated and expressed. There are different things about the epigenetics that are studied, but the more studied are pretty much the DNA methylation and the acetylation of the histones. Those definitely are very well studied. There are other things beyond that also happen in the RNA once that it's already translated, and it's this one gets destroyed or not. And I'm going to talk a little bit about that, but not too much, and then way more what happens after that. So there are two chromatins that we can call, the euchromatin and the heterochromatin. And when these histones get together and they form what we call the nucleosome, you cannot read any of the barcode. And this is called the heterochromatin. When they open, they go the euchromatin, and that's the part that is reading. But even if it's read, if you can read that barcode, something can happen to the barcode that it covers it. And that's what we will call the methylation. And the methylation doesn't let it read, therefore not translate, therefore not express. And this is important because we all have in our cells the same DNA, but we have cells that go to turn into neurons, and then we have cells that turn into skin cells. And the reason behind why this differentiation falls here in the epigenetics, but that's only on you. The same thing will happen on diseases and protective factors. You have two brothers, and maybe you have two twins with the same DNA, and they are exposed to different environments. One works out, the other doesn't, and they both have obese parents, so they have the risk factor of obesity. And why the one that works out doesn't get obese and the other one does? So that's also there are certain things, the environment that you are exposed to, in this case the intervention of physical activity, that will protect you from expressing what it's already written on the DNA. And this is other thing important, because 20 years ago we thought that what is in your DNA, it was set in stone, and therefore you cannot change it. And it's been changing in the last decade that no, we do have some work that we can do to avoid these things that are brought by our generations, and we can actually do something about it. So
this is hugely important, Jose, and I just want to ask a couple of clarifying questions to see if how I think about it is consistent with how you're explaining it, because this is a really important point for people to understand. And I think something that is near and dear to the CrossFit community's heart in that we have a lot of influence and self-control over how our genetic predispositions are expressed through our behavior and our nutrition and our environment and the stressors that we experience, right? So when we look at your diagram and we understand kind of what we're seeing with DNA and the histones, how I think about a histone and the purpose that it serves is essentially the DNA is using that as kind of a storage capacitor, right? So that it wraps around the histone so that we can pack these huge, extensive lengths of DNA into an appropriate space inside the cell. And then when it needs to be expressed, it gets unwound from the histone, correct? So that that portion of the DNA can be read and expressed. Is that right?
Yes, correct. So it's like the histone, the DNA is wrapped, covered, you are not expressed, but the histone itself can uncover and express.
Yeah.
But even if it's uncovered, we have still another methylation. Is it expressed or not expressed, this gene? This one can be protective or can be risky. And that's only two of the things. Then the histones is called the paracetylation. The acetylation would be the one that brings them close or open. The other one is called methylation. Those of the most studied of this one. And
yeah,
we
and
have more.
the other thing too, to the way that I think about it is that methylation, right, or the expression of our genes and what methylation does, where these methyl groups are, is essentially how we read our genetic recipe. And it's almost like reading a recipe, right? So if I'm reading a particular recipe to make, granola, and every third and fifth word is missing, or I'm missing a paragraph, right? The quality of the granola that gets made is going to be quite questionable, as opposed to if I'm reading the optimal or full recipe, right? The best form of whatever it is I'm cooking or creating is going to be expressed, right? And so methylation, I think about in that way, it allows us, when we are methylating optimally, to express our genetic information in the healthiest form. Is that a good way to
Yes,
think?
but sometimes the thing is that the expression of a gene can be, for example, something that helps you not to destroy the mitochondria. So that's
Mhm.
protective. But sometimes it's a gene that can express cancer.
Mhm.
So methylation covers it, you don't get the cancer. In the other one, uncovers it, you don't get the destruction of the mitochondria. So the same pattern can work in for protect or risk. It depends on what the gene does. What is the information?
Got it. So it can lead to expressing it, or it can actually keep it from being expressed, and both of
Yes.
those can be healthy. Yep,
Yes.
yep, that makes sense.
I was trying to, before getting this conversation, I was talking to other friends. Hey, I want to see if I'm explaining it well. So I came up with the analogy of the barcode, but he understanding different. He was telling me it's like you have a gun and you wanna cock it, and the gun, the bullet can come out or not, being the risk factor, but it's do you have the right finger in it? Do you have is it locked? Did you cock it or not? Is there a bullet or not? So there are different things. Maybe different people get it in different ways. So different analogies work for everyone. And the
Yes,
expression.
I like that analogy. And as we always say, right, genetics loads the gun, right? Epigenetics pulls the trigger, right?
Correct.
And there's a lot of influences that can lead to, as you alluded to, is there a bullet in the chamber? Do you have a finger on the trigger? What direction are you pointing? All of those
Right.
things, right? So I think it should be really empowering for people, right? Because to your point, so many of us, and we've heard this mantra about obesity and other things, right? That it's 70+% genetic and everything else. And people get it in their minds that this is fatalistic, right? That
Correct.
there's nothing that you can do, that your genes are your future. And what you're saying is no, through epigenetics, behavior, nutrition, recovery, stress management, you get to control how those genes are expressed. And so even if you have a predisposition towards a particular phenotype or disease, through how you live and train and fuel you can mitigate that potential risk. Is that correct?
Yes, and we're going to put it have a graphic that you can tell all the good things that you can do for, which is, spoiler alert, it's what the doctor will tell you to do. So it's like what doctors have been telling you to do, that's it. So I'm just gonna pass fast on the next one, because if this is complicated, the next slide is even more. Once once it gets read, it comes the translation, and it comes the expression, and goes to the mRNA, and all these steps can be stopped. So even if your DNA gets read and it's turning to mRNA, there are other parts that it's there's called the microRNA that can also block to express the protein, destroy the RNA. There are so many steps. So the point of this slide is just to remind that it was not, as we thought, straightforward. You have the gene, then you express it. No, there are several stops. There are several steps that your body will take to make checkpoints before you go. So before you go and express it. So in that matter, it's what we were saying again. If you have the gene, doesn't mean that you will have the condition. If you have obesity in your family, it doesn't mean that you will get it, the condition. There are other ways. And coming on the analogy, it was like, okay, you get bread, but does it get the information passed until you get the package at the end? There are several steps that you will have to go, and that's the reasoning behind all the lifestyle modifications that we do for everything. So the next point that I want to talk is the aging as a disease. And this was something that when I was reading, studying for the level 3, I came across with this study. And on the right, you see the VO2 max related to the age. And it's so interesting that on this chart at 40 years old, you have the same VO2 max of an endurance trained person that at 70 years old. So aging will decrease some function capacities, and we all know it. As doctors or as health practitioners, you are aware that we are a little more permissive on the blood pressure of the 80 years old male, or we are a little more permissive on the functional capacities. Oh, you cannot really run as before. That's fine. It's your age. Oh, you have anything we expect. The blood work comes as your kidneys are not in the best shape. We are a little more permissive. It's okay. It's part of the aging. But there is a culture wave that they are thinking that the aging itself is a disease, and as a disease we should treat it as any chronic disease. So it's what we were talking at the beginning about Healthspan and expression of the Healthspan to decrease morbidity. So the same thing that we have right now, the lifespan is 78 years old in the US, pretty much. And you have a person with high blood pressure, the lifespan will decrease to 45, half of it. So the kidney, let's take the kidney function. The kidney function that you expect at 80, you will see this person, if doesn't correct the blood pressure at 40. Therefore reducing the lifespan at half. So at this point forward, you will start experiencing the same complications that you see you would expect someone at 80 years old. But why if the 80 years old, as if we see the aging as a disease, why do we expect this person to decrease all function at 80 if with lifestyle modifications we can expand that health span to keep increasing, same VO2 max as before, or same kidney function as before, or same functional capacity, lung capacity. So that's why if you have a hypertension asymptomatic, you treat them with lifestyle intervention and maybe medication. Same thing would be like the subclinical aging. You are asymptomatic, but you still need to do lifestyle modification, training, diet, stress management, recovery, and that will also prolong your life and health span. So that's something that it's just starting to happen, that people seeing aging also as a chronic disease that we all experience, and therefore we all should get the lifestyle interventions that we do in CrossFit, diet, that we also. So now, let's connect epigenetics and aging. So there have been some libraries made by scientists in this field that they have found out some markers in epigenetic markers, and they put it as under the name epigenetic clock. What they expect to be methylated or not methylated in a 30-year-old, in a 40, in a 50. And then you can have two pictures. What is your biological age and what is your epigenetic age? So someone that has done everything to decrease their longevity, let's say ultra processed food, sedentarism, alcoholism, smoking, all of these things decrease your longevity and increase your epigenetic clock. So you may be 40 years old, no disease at this moment, but you consume ultra processed food, you smoke, and you're sedentary, your epigenetic clock will be 10 years ahead of you. So asymptomatic, normal, apparently no symptoms at this moment, no problems, but you'll be 40 years old, but you will have 10 years ahead at the level of your DNA. And this is done, but different things on the mitochondria, the ROS, or the increase in genomic instability, even the length of the telomere, every gene at the end there has the telomere, and it keeps cutting until it start cutting part of that. It's important to express. So all of these markers or biomarkers, we can see it now in their libraries to see what is expected. So you have this picture: biological age, and also the epigenetic clock, your epigenetic age. And through this is what I dived in to see the physical activity and the biological and the epigenetic clock.
And just to clarify there, so when we look at this, right, we're comparing, kind of contrasting two things. So we have our chronological age, right? So how many years we've actually been on this planet.
Correct.
And then contrasted with our biological and epigenetic age, which are, yes, I may be 50 years old, but if I've trained and I've eaten in the right way, my biological age, which takes into account a lot of things, but it includes my epigenetic profile, may be 40. Is that correct?
Right. That's the opposite is correct. You may be 40 years old, but because you have been doing the right things, then your epigenetic clock will be decreased. So yes, that's the opposite was too. So this is a picture from an image that it was shared on one of my research, and on that research was the lifestyle factors and the epigenetic modifications, and we try to get the pillars of health that we call in lifestyle medicine, which is There are 6 pillars of health, but we only dived into 3 of them, which is physical activity, the nutrition, and the stress management through some intervention. And this was a general research, and we found that all physical activity was improve all the mitochondrial effects and also decrease the stressing factors, therefore producing promoting the protection in that epigenetic. In the mental health, the stressing, we found that the mindfulness practices were the ones that elicit the most effect on the DNA methylation. And then on nutrition, we try to dive on all nutrition. There's not much more, not many information about it, but Mediterranean diet, the DASH diet were the ones that have been the most in studied. There is a lot of space for studying in this field, but those are the things that we found. In the physical activity, all physical activity gave the good effects on the epigenetic level, but the high intensity interval training was the one that elicited the most effect on the protection. So this is one of the general, and then let's talk about the physical activity and epigenetic only. And if I would like you to get something, would be these 4 things. The physical activity was the one that produced the greatest response of any other intervention, and the great response was on the DNA methylation. The DNA methylation is also one of the best studied, so there is some bias there, but DNA methylation and is the greatest response. The 3 physical activity studied produce benefits: the high intensity interval training, resistance training, and aerobic, the 3 of them. And then, but the high intensity interval training was the greatest response. And one interesting thing is that the sicker you are, you got the best response at the aging clock. These 4 things that we're talking about, the aging part. Some interesting things, like in CrossFit, we do all of these 3. We work on aerobic, we work on resistance training, we work on high intensity training in intervals sometimes. And if you rest a lot between reps, that's also considered interval training. So this is the part. Okay, so these are the epigenetic and aging. I just put a few of the reports that we found. For example, Stress and All found that the DNA methylation inhibits the mitochondrial death after 12 weeks of HIIT. Then Barris also found methylation in the adult skeletal muscle. This was a very interesting one, because what he decided to do is only one class of high intensity interval training, then did a biopsy in the leg, and right there they found already pre-HIIT and post-HIIT already found changes in the DNA methylation. But they did one more biopsy 12 hours later, and they found that changes were temporary. So even if you found changes in one session, this is the one that you need consistency to keep those changes happening in the DNA methylation. On the third one, same thing. The aging just in the epigenetic clock, these markers of the epigenetic clock after HIIT, it happened. The Lohmann was very interesting. After the HIIT, they did the transcriptomic age, which is the library itself, to see what is the reduction after 12 weeks after 10 interval trainings. They did 10 trainings, and they found that it was a reduction of 3.59 years compared to what it was before.
Amazing.
And then the next one also, these are all about proteomic changes and age related. And the last one is not about DNA methylation, but over there they review the length of the telomere, which is something that we lose with the aging until we start losing the gene itself. They found that it was reversed. So it's pretty much like rejuvenating the DNA, bringing it back. And they found that with aerobic endurance training, but also with intensity training. They didn't find it with resistance training, though.
Yeah, I mean, a really important point here to highlight, and I think one of the things that we hear a lot from individuals is that they have to get fit before they start CrossFit, or they're not in good enough shape to start CrossFit. And individuals who are the most sedentary and the sickest get the biggest benefit. And
Yes.
the beauty of the methodology, right, is the universal scalability. So that is a myth that we really need to continue to push against and highlight the fact that really the sickest people need this the most, and they get the biggest benefit from a health standpoint. And that the science, whether you look at their VO2s or their skeletal muscle, visceral fat, or now, right, their biological or epigenetic age, all of those things improve. And the people who have the worst get the most benefit. It's a beautiful relationship.
Yes, and right after that, they did this one. The sicker you get, the greater response. In this exactly, they decided to put people with heart failure and already fibrosis, which is, to me, when I started reading this paper, was okay, how did I get this done? But then after that, they found that it was reduced the fibrosis of this heart failure, these people with heart failure and the type of fibrosis, and also reduce the epigenetic. There's another one that there is also about the obesity in women, and obviously they improve all the biomarkers related to obesity. But in this group, there was severe obesity. They also found the best regression on the epigenetic age, and that's we've seen it. So all of these only backs up what we already seen it in the CrossFit space and in everyday, like the curve, the person that comes starting to get PRs and get better and better, and the worse you come, you get the best, at least the best benefits a long term. So yes, all of this information that I get to gather around was pretty much to back up what we already know, but we wanted show me the data. That's how we go in the Occident.
Yeah,
So
this study in particular, Jose, really caught my attention. I saw this when I was reading your paper, and that reversal of fibrosis, I think they saw a 10% reversal. And people may not understand how significant that is. I mean, you can think about this essentially as like scarring and change and hardening of the tissue of the heart. And what this study found was that it actually rejuvenated and reversed that scarring and fibrosis, and the hardening was there, which in heart failure, right, allows for improved contractility, blood flow distribution, and that's capacity, right? That's quality of life. That's being able to walk and move around with your grandkids. That's being able to go up a flight of stairs when you couldn't before. I mean, that is unbelievable. And to see actual tissue reversal that's measurable in that period of time is really impressive.
yes, yeah. So that's the greatest part of there, that you can have. It just not only what we would like to be only in the aging on numbers, but also on function too. So that's the part of like seeing aging as a disease, but the diseases itself that we can diagnose can be reversed, and we can see that at the DNA level what happens. So that's that's why we have. Let me. So if you have any questions or you want to dive in a little bit more.
Yeah, no, that was fantastic, Jose. I think that, the important message here, and it comes back to a lot of things that we talk about in CrossFit, is the ability to own your health, right? And to be the one steering the ship and to take charge. And I think when it comes to, our genetics and genetic predisposition and family history, having objective scientific evidence that shows that this isn't your fate, that through your behavior, what you do, and how you live, how you train, really can give you back control and dictate to what degree those genetic predispositions are expressed, if they're expressed at all, should be hugely empowering. Uh, and so I hope that, if nothing else, people take that message away from your presentation. And none of us should feel fatalistic about our genetic predispositions or family histories or what have you. If anything, it just makes training and doing what we do in the CrossFit space even more important, so that we can control how those genes are expressed. So I think that was a great presentation on a very complicated topic,
Right.
but it'll give people some frame of reference to understand, okay, well, what is this methylation thing? It's like, okay, well, that it essentially is the governor, right, with how my genetics are expressed and what parts of them are expressed, the good parts, and how the dysfunctional parts are kept from being expressed. So it's very, very, very powerful. And the great thing about it is there's so many influences with training, but nutrition as well. And it's meat and vegetables, right? And our veggies is highlighted in some of the scientific presentations that you highlighted with folate, and we know that B12 are huge influences, and choline as well, right? For methylation, which is more from lean cuts of meat, cheese, eggs, dairy, etc. So all of those things nutritionally have a huge influence, and a lot of us have genetic predispositions that increase our need for those micronutrients. And so the foundation and the base of our pyramid, right, plays an important role, just as the training does, in optimizing methylation. That's why the comprehensive nature of the methodology is so powerful, because it's touching all of those health pillars. So that's really, really great. Jen, do you have any questions or thoughts before we wrap up?
No, I loved it. I think it's important to just continue to shed light on the kind of scientific foundation, as Tom alluded to, the methodology being a great driver towards health. And we like to say that CrossFit is the gateway drug, and it just kind of then gets people to be ready to accept information in and around like the importance of this health journey and how they can own it as an individual. And so that's why I was excited to be able to have you on and talk about your study as well and stuff. So I thought it was great. Thank you.
The other thing I would say too is that, I know residency is a very relaxing time, and you have nothing going on. You have all this free time on your hands, but and I'm sure, I know you've thought about this because we've chatted some about it, but doing a prospective study, looking particularly at the CrossFit community, where, so many times we get, especially with a meta-analysis or a retrospective review, right? There's just a lot of different buckets and categories that get put in together, and we still see statistical significance, but it would be really fascinating to look and compare, a sedentary group looking at their biological versus their chronological age with a really good epigenetic clock, and then having them do 12 weeks, and then looking at all of those metrics later. That should definitely be on our short list. So if there are people who are listening out there at affiliates who would be interested in participating, we'll plaster Jose's pager number all over the presentation so that you can call him at 3 a.m. because he will be up.
I will be.
Because we don't get to sleep when we're in training. But I am really excited about that. I think at some point, hopefully on an elective month, or once you get through and can catch your breath again, that would be something that we can lean into. So I think that would be a really cool way to leverage the current science.
Actually, the reasoning of these systematic reviews that I've done, it's to come. I already exposed it, but to my program director, hey, I've done this. Now let's do it here. Let's
Beautiful.
put it here. And I've talked to a couple of CrossFits here. They are willing to be on board to provide. So hopefully not this year. They're still adapting, maybe in the future. But the voice it's already out there, and yeah.
Well, that's really exciting. Yeah. However we can support you, would love to make that happen. Well, I appreciate you taking the time out of your ridiculously busy schedule to be with us today. I look forward to reading your next research paper and seeing what you do in the future, and more so, just really thankful for your dedication to this part of medicine. You could have gone into any field and you chose to continue on in family medicine, leveraging your passion and your skill set to help people really understand their health and take control. And it's not the easiest way to go. And we all know that. And so I just want to commend you on, putting your money where your mouth is, and working with people in that way. It's a big deal. So thank you very much, sir.
Thank you. Prevention is the way. Prevention.
It is. All right. Well, have a wonderful day, and hopefully get some rest. Okay.
Thank you.
Okay.
Everyone.